perfetto.
Okay, good morning. Today we have Maria Elena Castellini that is going to speak. She's going to speak about her training path at CIBIO and why she decided to join CIBIO for her studies.
Okay, so good morning, everyone. I'm a PhD student, a third year PhD student at CIBIO. And so I've always thought that research would have been a part of my life since I started becoming fascinated with the world of biology, chemistry, biochemistry during high school. So I decided to follow my passion for science, and my university choice fell into the Bachelor of Science in Biomolecular Sciences and Technology, and then I chose the Master of Science in Cellular Molecular Biotechnology at the University of Trento. So during these years, I had also the possibility to do my both bachelor and master internships in the Laboratory of Neural Development and Regeneration at CIBIO, and my PI is Simona Casarosa. After the graduation, I obtained a postgraduate research fellowship, and I had the opportunity to continue with the project that I started during my internship. So during this postgraduate research fellowship, I had also the possibility to do a research experience abroad. So I went to the laboratory of Dr. Patrick Blader at the Université Paul Sabatier in Toulouse, so in France. And this was really good experience for me since I started getting in touch also with other academic realities. So I met new people, new also ways of doing science, because I think that every laboratory have a different way to do research. And for a researcher, it's good to have to get in touch with many people, to learn many things. And yeah, it was a great opportunity, both personally. So I met new people, also new cultures, but also professionally. So after the postgraduate fellowship, I started my PhD program, obtaining a university fellowship at CIBIO. And I decided to continue working and studying as a PhD student at CIBIO, since I think that the PhD program in biomolecular sciences at CIBIO is really well organized. There are many interesting courses, seminars, and I think that there is also an international and also stimulating environment. And last but not least, there is a lot of collaboration between people, and also different labs that work on different topics. And I think that collaboration in the scientific research is fundamental, is necessary. And yeah, another thing, I also decided to continue working on my project, because I was really enthusiastic about the project, the preliminary results, so I was really into my project. So this is my path, my training path. I'm still continue with my PhD program, and yeah, this is I think the main
reason why you
yeah, the main reasons why I chose to do the PhD at CIBIO.
And so why don't you tell us then your let's say day of work, what you do when you arrived and what you do in the lab, and maybe a little bit also about your project.
Okay, so normally on Friday I have a lab meeting with my PI to discuss about the experiments that I have to perform during the week, and so I can plan the experiments for the next week. And normally, since my project is focused on investigating some molecular pathways for retinopathies in zebrafish, I have to work with zebrafish. So normally twice a week or more, depending on the experiments that I have to perform, I work in the animal facility, so the space where all the animals are maintained, at least the vertebrate ones. And here, for example, I cross the fishes, I perform some microinjections, so some experiments. I also check how my fishes are. And when I'm not in the animal facility, mainly I work at the bench in the laboratory. And yeah, when I arrive, usually I have always a bachelor or master students to follow during their internship. So we plan all the experiments together, and we read the protocols together, and then we do the experiments. Regarding the working hours, I don't have a fixed number of hours per day, since the PhD is a fellowship. So depending on the experiments that I have to perform, the working hours change. Sometimes I work also during the weekend, sometimes no, it depends on what I have to do.
Sure.
And regarding my PhD project, is focused, as I said before, on investigating the molecular mechanisms underlying some types of inherited retinal dystrophies, such as retinitis pigmentosa, and possibly also find new treatments for these diseases. Currently, we are using zebrafish as animal model to study retinitis pigmentosa in vivo by modeling the disease in the fish. And zebrafish, a little freshwater fish, which has become during the years a tool also for the study of human ophthalmological disorders, since its visual system is really similar to the human one, and it resembles the human one better than other animals models such as mice. So yeah, I think that I know that you will receive also information about eye throughput screening, and zebrafish is also a tool to do this eye throughput. Since normally, if you have to test drugs, you can put one embryo per well, and then you can directly put your drug into the medium when the zebrafish, so the liquid where the zebrafish are, and then the drug is absorbed through the skin, and so you can test the effect of the drug directly on the embryos. And since the plate has many wells, you can test a lot of drugs. So it's a high throughput screening also for drugs.
Yeah, thank you for this nice description. So working in Drosophila, I also appreciated this other use of this model system to do screening, which is very important also for the biotechnology environment. So another question that I wanted to ask you is, what do you think about the relevance of the public opinion on some research discover or something? So this is something that maybe you will be exposed to if you are going to expose your research to the public. And so how much do you think that the opinion of the public counts on this?
Yeah, I think that nowadays there is a sort of gap between the community, so everyday people, and the scientific community. So for everyday people, sometimes it's also difficult to know exactly what we are doing in the lab, since there are some events that aim to explain what we are doing in a lab in a simple way. And I think that nowadays, so we saw, for example, with the pandemics, that there are many fake news about science. And I think that it's really important for scientists to communicate what we are doing in the lab to everyday people in a simple way, not so we cannot think that we can speak with scientific words which are very, very difficult and can lead also to misunderstandings with the community. But we can also think that if we explain our work in the lab in simple words to everyday people, maybe we can reduce this gap within between the community and the scientific researchers, which are seen for many people like strange people that are working in the lab 24 hours per day. So I think that it's really important also to reduce these fake news that can lead to many bad situations.
Sure.
And also, for example, I took part to Notte dei Ricercatori, which is an event intended to bring people closer to science, organizing some activities to explain science to the community, also with games or also with posters. But these posters are not the same that I bring, for example, to congresses or meetings. So are normally are funny, are written with simple words, so everyone can understand what we are doing.
Yeah, that's quite interesting what you're saying, because I think you are mentioning some words in your talk, like simple words, explain things in a way that everybody understands and so on, and which is really important for the students of this course, of course. And so the last question is, what is your message to the students of this master's? So what you want to say to them?
Yeah, mainly I think that the message is that nowadays we are living in a world where fake news seems to be a sort of fact of life. So I think that science is not immune to this trend, and it has a great impact on creating alternatives scientific facts. So the pandemics, as I said before, give us a great example for this, since there have been many fake news about coronavirus. So what I suggest is to communicate facts based on scientific evidence. So read papers, find data, and then communicate science, because fake news could lead to very bad consequences. And also adjust the terminology also to the public. So if you have a scientific public, so you have to communicate to scientific researchers, you have to use scientific terminology. But if you have to communicate science to everyday people, don't use terminology which can confuse the general public.
Okay, that's very clear and strong message. Thank you. So I think we are finished today. And if you would like to participate to the discussion with the students and the other people who are interviewed, is going to be the 28th in the afternoon. I think after 4:30 in the afternoon. That's our time so far. So, and I will send you an email for the invitation. Okay. Thank you.